Right, hello everyone, and welcome to Lecture 8 in the ABI African Bioinformatics Institute and African Farm Breeders Association joint webinar series and a hackathon. So in Lecture 8, Nelson will be going through data formats and file formats that you need to use. So the success of genome-wide association studies, or GWASs, and genomic selections depends on the quality of the underlying data. So poor quality control decisions can made early in the analytical pipeline can propagate downstream through analysis, leading to inflated associations, reduced prediction accuracy, and ultimately unreliable breeding decisions. So in this webinar, we'll provide a practical guide for data quality control, file formats, and conversions for GWAS and genomic selections. And we'll cover essential QC steps, your quality control steps for genotypic data, including filtering for missing data. So we're very lucky to have Dr. Nelson Lubanga, who is joining us today. He's a quantitative geneticist at the Institute of Biological and Environmental and Rural Sciences at Aberystwyth University. He holds a PhD in plant breeding and genetics from the University of Nottingham and has over a decade in experience spanning academia, industry, and international research, including positions at University Kenya and the Rosen Institute and Duman Orange. Nelson's research focus on genomic prediction, breeding program optimization, and accelerating genetic gain in crops that are facing challenges of climate change and global food security. So over to you, Nelson. Thank you.
Oh, thanks, Sumir. Hello, everyone. So as you've been told, my name is Nelson, and I'm a quantitative geneticist by training. And this is part of the application of quantitative genetics series. So I'm lucky to be working with Professor Alexander, who's also in the among the audience. So today we'll turn up to what's perhaps the least glamorous part of the genomic study, but also among one of the most important one, which is quality control and the file formats that surround it. So, and by the end of the session, we hope that you'll be able to take a raw file of genetic variants and turn it into a clean, reliable data set that you can trust for both GWAS and genomic prediction studies. So I'll start by showing you where we are going. We'll begin by placing.
Sorry, Nelson, quick point. There's another slideshow on top of, yeah, there it's gone. Okay, perfect.
I don't know why it keeps on coming. So we'll begin by placing quality control within the wider analysis pipeline, so you can see exactly where it fits. We'll then talk through how to choose a genotyping platform for your particular question and how to read the main file formats that you'll encounter. From there, we'll move to the heart of the session, which is genomic quality control, and then phenotype quality control, and throughout the thread that ties everything together is reproducibility of the analysis. So the big picture is everything we do today rests on the single idea. Reliable analysis starts with reliable data. So what you put into the model is what you get. So if you put in poor quality data, you get poor quality results. And every genomic results you will ever produce, whether it's GWAS or genomic prediction, it's only that it depends on the data that you kind of put into the model. So before we start, it helps to see the whole journey from the wet lab to the bioinformatics and the quality and quantitative genetics. So in the wet lab, DNA is extracted, libraries are prepared, and the samples are sequenced. In the bioinformatics pipeline, those raw sequence reads are quality checked, aligned to a reference genome, and turned into called variants, ending in a VCF file, and then in a quantitative genetics, which is where we'll spend our time today. We'll filter and impute the variants, examine population structure, and build the genomic relationship metrics, and finally run some GWAS and genomic prediction to inform some breeding decisions. Um, so, so where does the QC fit in? Um, so quality control fits at the very specific point in this pipeline. So after variance calling and before any analysis, and that position is really the whole message. So everything downstream from the association test, breeding value estimation, um, inherits the quality of whatever QC allows through it. So why is QC negotiable? So the honest answer is that raw SNP data are inaccurate and incomplete. So errors creep at many steps, from sampling errors, from plating through to the genotypic itself. Sorry. And if we skip quality control, we pay for it in three ways. First, we could get false GWAS signals, which could inflate both false positives and false negatives. Second, we could get biased breeding values because a poorly constructed relationship metrics and unadjusted phenotypes could distort and the estimates and the rankings of the selection candidates. And third, we risk drawing the wrong biological conclusions. And this is very common in candidate genes analysis. So here, this is courtesy of some of the work I'm currently working on. So it's kind of good to have an overview of the overall marker distribution across the genome. So this is one of the crops I'm working on at the moment. And so this figure gives us a brief eye view. What you can see immediately is that the coverage is uneven. Most chromosomes are well covered, but a few here, the chromosomes 3 to 6, show some large gaps where the markers are sparse or absent. So these gaps could be most likely repeat-rich regions where short-read markers simply couldn't be placed during the SNP calling. So probably this matters because some of those regions have limited power to detect associations or capture relationships. So we could interpret genome-wide coverage within that limitation in mind. And then sometimes you ask yourself, so what kind of genotyping platform should you choose? So reading from the top, the marker density climbs steadily from the targeted assays like CARPS, which gives you a handful of markers for validation or routine screening, up to the SNP arrays and genotyping by sequencing, and finally the whole genome sequencing, which gives you everything. So one method worth flagging out is the DATSeq, which doesn't need a reference genome, making it valuable for orphan crops, for instance, which usually don't have reference genomes. But the single most judgment is this. So you should match your SNP density to how quickly linkage equilibrium decays in your species. So for the outcrossing species, where the LD breaks down very fast, so you need very many markers. And for the inbred or selfing crops, where the LD extends very long distance, so you need much fewer markers compared to the outbreeding species. So we'll move to genomic data formats, file formats. So we have the VCF and its binary sibling, the BCF, is one of the universal exchange formats for storing variants within full flexibility. So we have the PLINKs binary trio, the bed file, the beam, and the farm files. Sorry. And this is what we commonly use for quality control. And then we have the HapMap formats, which we'll use for tools like in GAPID or in Tassel. and also the numeric formats we commonly use in genomic prediction and GWAS. So this is how a VCF file looks like. I don't know if you can see. So we have the VCF header, and then the columns of the VCF. So for the column, we have the chromosome, the position, the ID, the reference, the alternative, the quality part, which shows if it's passed or not passed the filtration. We have the information file, the format, and the sampling part. So after this presentation, I'll be working mainly with PLINK. I like working with PLINK because it kind of has so many functionalities, but you can use other softwares if you like. So first of all, we'll have to create PLINK files, which is the bed, beam, and the farm files I just shown you. And we use a simple command. So if you're starting with a VCF as the input, so you kind of just say make bed and then out. So this is what will come out of the PLINK file. So the HapMap format is another format that you'll come across, particularly in association analysis. It's simple, human readable, and each SNP is a row. Each SNP row is a SNP. The first column describes that SNP, and the remaining column gives the genotype call for each sample. So it's less compact than the PLINK binary formats, and it's much more readable, and much makes it convenient for tools like Gapid and Tassel. So we also have the numeric formats. So this is the simplest representation of all. So each genotype is recorded as 0, 1, or 2, and counting the number of copies of a reference allele. And so this is what most genomic prediction packages expect because it feeds directly into the mixed model and relationship matrices where they build. So overview of the matrices. So think of this as a pipeline on its own. So we start with the raw SNPs and then apply each filter in turn: missingness, minor allele frequency, Hardy-Weinberg equilibrium, heterozygosity, LD pruning, and before finally imputing any remaining gaps. And over the next few slides, I'll walk you through how each of these in turn, and I'll explain just how to apply them. So for the missingness, so we have per sample missingness and per SNP missingness, and we have a command that we use this in PLINK. So the idea is straightforward. We want high call rates from the SNPs. So typically above 80% or 90%, both for SNPs and for each sample. Sorry. So a SNP that fails to genotype in many samples, or a sample that fails across many SNPs, is unreliable and should be removed. So for example, if I put this threshold, I mean, I want to remove SNPs that have like more than 10% missing data per sample. And also this one, I want to remove about 20%, samples with more than 20% missing calls for a SNP. I'll show this practically at the end of this presentation. So next is minor allele frequency. So this is simply the frequency of the rarer of the two alleles at a SNP. So very rare alleles carry little information in most GWAS and genomic prediction studies. So for the simple reason that too few individuals actually carry the allele. So with so few carriers, the statistical power is low and the estimated marker effects are unstable and prone to genotyping errors. For this reason, the SNP minor allele frequency below the 5% are typically removed, which improves the power and stability effects estimates. Then we have Hardy-Weinberg equilibrium. So a large deviation from Hardy-Weinberg equilibrium can mean that one of the two things. It can generally be biological, reflecting selection or population structure, or it can be technical, signaling a genotyping error. So, and this is the crucial point for this work. For instance, in a selected or structured breeding population, and especially in an inbred or clonal crop, departures when you depart from the Hardy-Weinberg are often real and often expected. So that's why we need to do these kind of filters. So LD pruning is also one of the quality analysis we do. It's not standard in most analysis, but sometimes we'd want to kind of remain with markers that are not redundant in a genome. So we'll only want to work with markers that are, let's say, are not overlapping and are carrying redundant information. So in this case, we want to reduce the number of markers and in this case also reduce the cost of genotyping. So lastly, we want to kind of combine all the analysis in one. Instead of just doing a single analysis, let's say removing the markers with this threshold or minor allele frequency, we kind of put everything in one single command using PLINK. So you will call the B file this threshold for the number of so like Geno 0.1 will remove SNPs with more than 10% missing calls. That is per SNP missing filters. And then we have mine 0.2 remove samples with 20% missing markers, and then 0.05 minor allele frequency. So this will write the filtered file into bed format, and that's what PLINKs work with. So I'll stop sharing this. I don't know if there's anybody with any question for now.
If you have any questions, you can raise your hand and ask Nelson, or you can type it in the chat. I see Enoch has his hand raised. Enoch and Mary. After Enoch, then Mary. Please go ahead.
Thank you very much, Nelson. I wanted to understand on missingness. Are those like standard, or you can set your own thresholds?
Yeah, you can set your own threshold. I mean, if you don't want 50% or 60% missing markers. It's just you just put into the command. You only want markers that are not more than, let's say, 10%, or you can even say you don't want any missing markers in your analysis.
All right, thank you.
Thank you. Mary, please go ahead.
Okay, thank you very much, Nelson. I saw a slide where you presented the full view of the genome, the distribution of the markers. So what do you do with chromosome 3, 4, 5, and all of those that don't have? Do you actually cut off that area? What do you do at the end?
So that's a good question. So depending on your analysis, so if you are doing a genomic prediction, GWAS analysis, first of all, it gives you like an overview. You kind of know you won't expect any let's say candidate genes, or you won't expect any more significant markers in those regions. So it kind of gives you like an overview of what you expect in your analysis. So then you can make a choice if you really have very interesting areas within those regions. Now you can decide like you know that you have some limitation that you have missing markers in those regions, and sometimes it's not because that is because of wrong quality control. No, it could just be because the markers were not called in those regions. So that's the reason. So I mean, so
Okay,
it's yeah, so there's no like proper answer for that, but it's up to you now to decide what you want to do, and you kind of know is I mean, what do you want to do you want to proceed with that, or you want to kind of make sure that you can even go ahead and genotype more with other methods, like let's say whole genome sequencing, or kind of narrow down into that region and resequence again the genome. Just depends on your objective.
thanks, sir. That means you know that you have a prior information that that area there are you are
Yes.
interested in that area. Okay.
Yes.
Okay. Thank you so much.
Yep.
Great.
So,
Yes, Aaron?
any other question? So are you able to see my screen now?
Yes, Nelson, we can see your screen. Maybe if you want to make your font a bit bigger.
Uh-huh. Is it much better?
Kind of looks the same again. Maybe just stop sharing and try sharing again. Okay, try again.
Give me a sec. Is it much better now?
If you could
We
share
can't
your
see it yet.
Okay, sorry. Yeah.
yeah, much better.
So I have 3 files. I like working with this because I can move around if I'm working on Python or in R, it's kind of easy. So I did some simulation of a trait. So 200 individuals, 8 chromosomes, and with 25 chromosomes, so QTL per chromosome, which is about 200 QTLs, and about 7,500 SNPs per chromosome. So I was targeting about 60,000 SNPs. So this is just the simulation parameters I was using, and I wanted a complex trait. So we have the mean, under some mean of 2,500. We have additive genetic variance, and then G by E effects, and of course, residual effects, and is in 3 environments. So I will give out this slide, this script if I want, if you guys are interested. So, but the aim was to kind of come up with phenotypes and genotype data that I can use for demonstration for this particular series. So so the SNP, I'll just click on my left. So the SNP will, the VCF file will look like this. As you could see, I could see in the presentation how you found the meta data sets, the meta information looks like this. The phenotype file will look something like this. Just reduce that a little bit. So we have the genotype, we have the environment, and then we have this is the genetic value. This is the phenotype. Yeah, so that's basically what we want to do. So for the QC, so the end goal is to kind of develop the bloops, the blabs and the blues for use in genomic prediction or GWAS analysis. So I normally use ASRemol, and for visualization, of course, it's ggplot2 to kind of just visualize what you do with it. So the first thing, of course, is to call the file, and then look at things like, is the files, are some of the columns missing? Kind of a simple line of command. Then you kind of just make into factors because that's how ASRemol works. So all this is just kind of some quality checks. So then you'd want to look at things like the percent missing values, the number of missing values, and things like the mean, standard deviation, things, so kind of the quality, some of the summary parameter statistics. Then you want to see how each environment is represented. You'd want to see things like how they spread in each environment, all through this. So you'd like to see the box plots of how the data is kind of spread across each environment, the histograms, the QQ plots, to kind of see if your data is kind of has many errors or not, the outliers in the analysis. Yeah, so I've made this analysis, and then at the end of it all, we have how you kind of estimate the blabs and the blues. So for the blues, you want to make sure that the environment is treated as fixed effect. So this is your model. I mean, you can make the genotype as fixed effect, and you could also make the environment as random effect in the model. But for the blabs, here you're making the genotype as random effect. Depending on what you want to analyze, you could also make the environment also as a random effect and the mean as a fixed effect. So just depends on what you want. Yeah. So I'll run this analysis. So if all this analysis I can just run by this click of command. And on my right, you can see we have some, let me just bring it like this. So you can see, if you're able to see like this, so this is like how the spread of the phenotypes looks like. So we have so these are the box plots. So these are the distribution, the frequency distribution. We can also like visualize the QQ plots. If you want to look at the potential outliers, you can kind of see if we have outliers or not. If you want to see how the blues and the blabs kind of make that comparison in a plot, you can also visualize residual versus the fitted values. You can always visualize this. So if you're interested, I can always share this script. And then the last one would be the
Nelson, sorry, we have Tinashe has his hand raised. So Tinashe,
okay.
to ask a question.
Oh, sure. Thank you. Can you hear me?
Yes.
Thank you so much, Nelson. I wanted to find out because you are talking of ASREML, which is a paid package.
Yes,
So
yes.
do you have any other alternative packages that we can use that might, since some of us might not have access to the paid packages?
So yeah, we do have other other free packages. Which can come top of my head is okay. Why I like ASREML is because you can run both genomic predictions. You can fit your analysis in whatever way you are. It's very flexible, and then it's very fast. So if you're dealing with millions of data points, it's so easy to kind of use ASREML compared to other packages. But if you don't have ASREML, I would recommend I'm trying to remember. Okay, maybe if you can email me, because I've got some analysis I did with some packages. If you can just email me, then I can take you through some of the packages you could use. But if you're working for an institution is able to kind of subscribe to ASREML, I think it's one of the best softwares to recommend.
Okay, I'll send you an email. Thanks.
Yes, thanks. So lastly, I will just take you through the genotype analysis. which should be, you should see a file like this. I'll just make it full screen. Yeah, I hope you can. Let me try to increase the font. Yeah. I hope the font is now clear.
Yes, it is, Nelson.
Perfect. So now I'll take you through some of the tools or some of the tricks we use for genotype quality filtration. So I use two main packages. There's BCF tools and PLINK. So BCF tools is mainly just to kind of, if you have a VCF file, kind of just see, kind of visualize or kind of see what is inside the analysis. So for instance, if I use this command, BCF tools view this, if I just copy and paste this here on my, yeah, sorry. Just give me a sec. So if I run the same file, it tells me I've got 60,000 markers. And if I run this file, if you just want to see like the head of the, visualize the, how the like your, the genotypes you have, or the varieties or the accessions you have, you run that command. If you want to see the first 5, this is the command you use. If you want to see, let's say, the chromosome, the position, the reference, and the alternative alleles, you could run this command here. Just copy it. Copy this. Yeah, so it can tell you, like, this is like your genotypes. This is the SNP. This is the alternative alleles and the quality filters. If you want to display the names of the names, you could use this kind of command. Don't worry, I'll share all these scripts so you can play around with it. So this one tells you like you have 200 genotypes. So this is just to kind of see what is inside your VCF files. And so then we now use the PLINK format. Now, PLINK now is the one that actually helps you to kind of do the quality control.
Nelson, move on to PLINK. I see Mary has a hand raised. So Mary,
Okay.
do you want to ask a question before we move on?
Yes, thank you. Are you on Arrow or on Ubuntu? Because I want to know.
So this is a command line. It's a bash. We call it bash or
Okay.
Linux. So if you use Linux or bash script, this is it's okay, I like using it because it's flexible. Again, we do have packages you can use to kind of visualize your VCF, but they're not flexible like learning some command scripts. So it's kind of like some kind of like a steep learning curve, but it's kind of worth learning some of these tricks. So once you have some knowledge of how to run some bash scripts, so it's kind of just easy to kind of copy and paste. So for instance, so this is PLINK. So I'm calling PLINK, and I'm telling PLINK that I'm expecting a VCF file, and this some other lines are kind of standard, like double ID, allow extra chromosome. So allow extra chromosome is, you're telling it like, this is not a standard human genome, it's kind of other species. So that's what it's expecting. And they're telling it to make into like a bed format. And then the output to save it as here. So when I put it like this, so this is like the PLINK file. I kind of created a file called PLINK. You can see here. So where it's going to save all this information. So if I copy all these, copy, and then paste this here, just clear. So it tells me that it's so it tells me that it has received 600 variants, and it has 200. So it calls them people, but because it was originally meant for the human genome, and then so 600 variants have all passed the QC, and we have 60,000 markers have passed the QC, and 200 individuals have passed the QC too. So, and they've been converted to the bed format, which PLINK's like working on. So if I want to calculate, let's say, the allele frequencies, so this is the kind of short command you put. So you call it PLINK. It's expecting a bed file. So this is the bed file, and then you just tell it to kind of allow extra chromosome, and then you have the frequency. So you're telling it to calculate the frequency of each allele. So if I copy this, yeah. Yeah. Yeah, and I forgot to mention I'm working with Visual Studio Code. Because I also find it more flexible because I can always move around Python and R in my analysis. And also it's kind of has got so many functionalities, much more powerful than let's say R or the standard terminal. So here it tells me that we have a genotyping error rate of 0.94, and the allele frequency have been calculated. So this is what I'll use to kind of filter in terms of minor allele frequency. So if I want to check, let's say, the allele frequency, I can just copy this and just clear this. So you can see, so we have the SNP, we have the alternative allele and the other allele, then we have the minor allele frequency of each SNP, and then we have the number of, yeah. So if I want to filter, I'll mainly focus on this column here. And then the other one is missingness. If I just click here, we want to see the number of missing markers per chromosome or per individual. Just run this. So then this one tells you like it has all done the analysis and has got two main files. So we have the IMIS and LMIS. So if I want to view all of them, I can just copy this. So you can see, so this is like the number of missing genotypes calls per individual. So you can see this is the percentage, sorry. Yeah, this is the so we have the IDs, and then we have I don't know what this one. But now this is like all the genotype. The most important file, the most important is this one and this one. And then we also have this information here for the LMIS. If you just copy this, paste, it gives you the same information. But now, let's say if you want 10, you don't want individuals with more than 10% genotypes, you kind of just copy this. So when you run this, so it tells you, for instance, so it kind of received 60,000 SNPs and 200 genotypes. And then here it tells you that the total genotyping rate for, so 185 genotypes are the ones that remained after the filtration. So meaning 15 of the genotypes were removed. And so this, it gives you all these kind of statistics. The other one is, if you want, let's say, to remove so if you want to follow, let's say, minor allele frequency, you kind of just copy this and paste it here. Yeah, so here it will tell you that so 15,000 SNPs were removed due to minor allele frequency threshold. So you remain with 41,000 variants. So it kind of gives you all these statistics. That's why I like using PLINK. And then if you just want statistics after filtering, for instance, you can just look at this, run this few lines of command. And it can tell you, for instance, so so this is the file that that has been written, and it can give you, it can tell you it kind of give you like a report of what you expect from that kind of analysis. And then if you want to prune the LD, for instance, so if I have another script for if you want to, let's say, prune the LD using this one. So you want to kind of prune within this region, 50 region, and then it's kind of after 50 it jumps to 5, and then the R squared that is kind of using to filter is 0.2. So R squared is like the measure for linkage disequilibrium. Let me just copy this. Yep. Yeah, so it kind of gives you all these kind of statistics and what you're looking for. Yeah. So lastly, because of time, so you'd want to kind of convert your scripts into 012 format, because right now, if you look at this script here, it's in VCF format. So this one will not help you in genomic prediction or GWAS studies. So you want to convert it into the 012 format. So you just run this kind of script after you filtered. Yep. So in this case, it has saved this file into snip 012. So if I click, it has saved here. So it's give me a sec. Yeah, so if I now give me a sec. Yeah, so if I'm able to look at how the file, the converted file looks like, it looks like you just run this command, and you can see everywhere is 0, 1, and 2. So instead of the PLINK format is now 0, 1, and 2 and which you can use for genomic prediction. And we have some few missing markers, which is written as NA. Yeah. So assuming that we have looked at the VCF format, and then we've converted the VCF to the 012 format or the numeric format. So there's also another way that you can, because some people just like using tassel. So let me just share this here. Yeah, so can you also see tassel? So if you want to kind of convert, let's say, the VCF file into HapMap format, most people also will use the tassel, and it's pretty easy. So you can just go to File. Okay, just open, depending on yeah. So this is the VCF that I'm targeting, and convert the VCF to HapMap format, because that's what we'll use for for GWAS studies that Alex will be running in the next coming few days. So this is like the format. So if you want to save it now, you kind of just go save as. Give me a sec. So you want to save into HapMap format, and it gives you, so if you go to this down, you can save into whatever formats you want, into PLINK format, VCA formats, HapMap. So in this case, you want HapMap format, so you just click on HapMap, and then where you want to save it, for instance. So I want to save it in a particular folder, let's say. Yeah. Yeah, so assuming I want to save here, I'll just say SNP one, for instance, and save. Yep. So that's how you save the files. So I still see I've got about 10 minutes of questions, if there's any question. But so that's how we, so don't worry about everything. If it's very overwhelming, don't worry about it. I'll share the scripts. But again, maybe if you're not very familiar with the command line tools, so you might need to maybe invest some few minutes to some few hours to learn about the command language, the bash scripts. Also, we have some tools also in R you can use for the manipulation. Yeah, but of course, if you're a beginner, don't get intimidated by this. It's kind of a learning curve for everybody, but I think it's something worth investing your time with. So I don't know if you have any questions.
Please raise your hands and you can answer any questions you'd like.
But also, if you want to kind of learn more, I think if you applied and you're able to come to South Africa, I think you'll learn more practically on how we do this, and also have some hands-on skills on how we can analyze your data set. So yeah, that's it.
Now, something you can do in the chat is maybe if you want to attach the scripts in the chat, then you can send the scripts through, or if you've got a GitHub repo, you can paste the link in. We have a question from Johannes Gabriel. Johannes, would you like to unmute and ask your question, or should I read it out? Okay, Johannes is asking, what is the purpose of pruning?
Yeah, so it depends on your study. Like for instance, recently I was doing a study on chickpea so I wanted to kind of see how we can save cost. So instead of using, let's say, 500,000 markers, because that's expensive for a routine breeding program, with only one, and most of the markers are have got redundant information, or are not very informative. So you'd only want the markers. So I wanted to kind of filter the markers that, using the LD approach, what you call the LD pruning. So this kind of avoids those kind of redundant SNPs. So we are only focusing on the SNPs that we believe are more informative based on LD threshold. So in this case, I was using LD threshold of 0.2. So I was comparing 500,000 markers with about 9,000 markers after pruning. So, in a practical breeding program, you see, you can save a lot of money instead of using all the 500,000. Because if I send the markers to the genotyping company, they'll charge me per sample or per marker, depending on whatever agreement you have. So, so in case I tell them I only need like 9,000 markers, but based on those LD prone SNPs, I'll have saved the cost significantly. And what I found is that the predictive ability for genomic prediction wasn't very different. Of course, we expect a lot of genetic variance between the whole genome sequenced markers compared to the LD prone. But I didn't see a significant difference between the two marker platforms. So that's one of the reasons you could use the LD prone markers instead of just whole genome wide markers. Yeah, again, depends on your objective. So
Okay, I see Nelson has shared the scripts in the chat. So if people want to download them, please go ahead and download them. Do we have any questions? Yes, Nelson, please go ahead. If you have any questions, you're free to raise your hands and ask them, or you can just type it in the chat and we can read them out.
yeah, and I encourage you to also register for, Alex will be running some other webinars from, is it next week? So I encourage you to also attend those webinars. It's kind of like a carry on from this. Yeah, and then the last one I'll be running will be the application of genomic prediction in August. So I encourage you to also register for all those webinars. And this one was just like, I mean, quality control is one of the, I was discussing this with Alex. So it's for a quantitative geneticist, we don't kind of routinely do quality control because we believe that the genotyping companies have kind of run through the quality checks and this, or the bioinformaticians have done this. So, but I was just sharing with him is like that some of these things I've had to learn. I didn't go to school to learn this, but I've had to learn this on my own because once I receive the data sets, it's not as clean as you expect. So, and you are kind of required to manipulate it yourself or kind of just have a sense of what the data looks like before kind of going down into the real analysis. So and it kind of helps to kind of understand this kind of your data sets before going into the quantitative genetics analysis.
I totally second Nelson on that. As being a bioinformatician and a data scientist, a lot of our work is actually doing a lot of QC and cleaning the data because once you're running through your analysis pipelines, you want to be sure that the results you're getting are true results and not artifacts of the data that's not being properly QC'd or cleaned.
Yeah, that's true. Yeah.
Do we have any questions, or would people like to raise their hands and ask any questions? Johannes, please go ahead.
Thank you. My question is related to the number of SNPs. I have a VCF file almost with 2 million SNPs. The number of accessions are around 400. I was trying to use RStudio to do some filtering with different tools like Dart, R, PLINK, etc. VCF tools. When I come to the RStudio, I'm using my server actually. It has got 256 GB RAM.
Yeah.
And it takes a lot of time to do even one process. It takes more than 1 day, etc., etc. So I was suggesting to reduce the number of SNP by pruning or filtering. I try to do practice these things by myself. And when I do some filtering, it comes to around 500,000. So it was a kind of a PhD student work. I suggested to reduce this SNP number as much as possible, but the student and his advisors are not willing to do that because they want to use all the SNP values. So what do you suggest? And this is one question. The other thing, can I get your email so that I can contact you further?
Yeah, that's fine. So okay, I had the same experience with chickpea data set. So they had done whole genome analysis. So and they kind of gave me about 3.9 million SNPs. So, and because I cannot work with 3.9 million SNPs, I try to prune using the standard methods like, let's say, 0.2 missing markers or 0.2 missing individuals, or sorry, and then minor allele frequency of 0.05, the standard code QC. I also got almost the same amount of markers you're talking about, 500,000. Still, I mean, you could still work with them, but it will take a lot of computational space, or unless you're working in HPC or cloud, it's fine. But if you're working in a standard MacBook or laptop, it will take a lot of computational space. So what I would suggest is, so what I did is I tried to kind of filter the markers. So number one, I kind of made the threshold very stringent. So instead of, let's say, saying 0.2, you could say you don't want any missing markers, for instance. Then you see how many it will give you. Or you say, instead of like saying you want 0.1 missing markers, you can say you want 0. So in PLINK, you could say the mind part, you could put 0, and then the genome part, you could also put 0, and then you see how much you'll get. So in this case, you only have full marker sets with no missing markers. You'll find that by kind of making those parameters stringent, you could reduce the markers significantly. Yeah, you can look at this if you want. You can send me an email. Let me just write my email. Yeah. Yeah, it's just try to play around with more of those parameters. But I mean, I can tell you it's not practical to work with 500,000 markers in your desktop unless you're working on a cloud. Yeah.
Thank you very much.
Okay.
Great, thank you. Maybe Nelson, what you can do is you can ask your university or your institute if they have a server that you're able to use, and ICTS can get you onto that server to run your analysis. Okay, do we have any other questions for Nelson? Okay, we'll give it a minute. We're 1 minute over time. So I've put in some links for the next set of lectures happening next week. So please do register for that. Professor Lipka will be covering GAPIT, which is an R package, which people can use for GWAS, especially in the agricultural sector. And with that, I guess it's just to say thank you so much, Nelson, for this excellent talk. And thank you, and see you guys next week.
All right.
Bye.
Thank you. Bye.
Thank you, Nelson, and everyone.